From AI to CRISPR, how should emerging reproductive technologies be regulated? The sponsors of this evening's event are the British Fertility Society and Merck, and PET are extremely grateful to them for their support. You'll be interested perhaps to hear that the Human Fertilisation and Embryology Authority have been thinking about how to update the Human Fertilisation and Embryology Act. And one would think that the sort of emerging technologies would be something that they would be considering in their conversations about doing that. At this evening's event, we're going to have an excellent panel of speakers explore the different technologies and their regulation. It gives me great pleasure now to introduce this evening's first speaker, Dr. Helen O'Neill. Helen is Chief Executive of Hertility Health, and she is also a lecturer in reproductive and molecular genetics at the University College London's EGA Institute of Women's Health.
So you'll see I've taken the liberty to add from femtech to AI to CRISPR, because I'll be starting with femtech. And though this is a term that bothers me somewhat because I think it underplays the technology that is required across the board in women's health, nonetheless, it's a good catch-all phrase that can be used, especially in this instance where we talk about emerging reproductive technologies. So my conflict of interest, or my disclosure, is obviously a major one in that I am founder and CEO of Hertility, and it's Hertility that I'll be talking about tonight. Moreover, in creating a technology that serves reproduction, I've learned about the regulatory processes, the technology gaps, and the need for proper communication with those for whom technology serves. So I'll be talking about this through the lens of an academic and also through the lens of a company and the lessons that I've learned along the way. But firstly, within the context of reproductive health, or certainly under the auspices of BioNews and PET, we're usually talking about regulation in terms of assisted reproduction. We're talking about regulating different practices that aid in assisted reproduction. And actually, when we look across the reproductive health landscape, from healthcare to fertility to menopause, there are many different areas where we'll see that technology will be aiding and is actually necessary in these different areas outside of assisted reproduction, and where we'll see that actually moving landscapes in terms of our societal expectations and our shifts within society for acceptance of certain things, we're actually moving towards, in certain circumstances, a total reliance on technology for reproduction. Moreover, when we start to see the sheer pressures that our healthcare system are facing in terms of the numbers, actually we'll see that technology is very essential when it comes to implementing and assisting healthcare systems and healthcare providers. So when we started to look at how we can transform reproductive health, we started with fertility. And we very quickly realized that actually it's very complicated when you look at the underlying conditions and things that could start to affect one's fertility. And when you need to take into account many different reproductive health conditions, you need to start to look at then the prevalence of those conditions. And it makes you realize that in trying to build a predictive algorithm for a fertility, for women's health, for individual conditions, the molecular determinants, the societal determinants, and every other kind of determinant that would lead to somebody's prevalence for a condition actually has to be taken into account. So what we started to see with this was that this landscape, though we look at technology as it appears within the context of assisted reproduction, actually technology is very much needed much further upstream from that. When we look at the statistics in and around women's health, or rather fertility or infertility, we see that actually one of the key causes or reasons for somebody attending a fertility clinic is that they are of advanced maternal age. And so education at a much earlier age would help in those technologies. So we also started to look at the patient journeys, and that was a massive educator to us in terms of what are the areas of legislation that need to be looked at when creating a solution or a tool across those different areas. But furthermore, what are the individuals? So when we think about treatments, it's funny to say that as IVF treatments don't treat the patients, that many of the technologies that we're regulating or the treatments we're regulating do not treat the underlying condition. They serve to essentially circumvent a condition. And when we looked at the different types of patients, we started to see that the field of assisted reproduction and IVF is a very rare use case, in that it is one of the only fields of medicine the patient doesn't have to necessarily be sick, and that the patient can actually request their treatment. It's probably one of the only areas that new technologies and processes have been implemented and then retrospectively analyzed through RCTs, as opposed to the typical model where everything is done in the lab and then brought and then tested on animal models. And so it's a strange but rare use case of how we can actually retrospectively look at regulation and see how things could have been done. And I think the examples that we'll see going forward, for example, with regulating for germline genome editing, are that we actually have the opportunity to regulate in a controlled way and not experiment, but analyze results in a much better way, and that in a chance that we actually didn't seem to take with IVF. Now we're seeing massive data coming out about looking back 10 years after the IVF cycles or egg freezing cycles, but really that should be prospectively built in to models whereby clinics are responsible for research that's ongoing. So we actually, for some of our early data shows that in building a diagnostic test that enables us to look not just at data that is from women and men approaching a fertility clinic and therefore are of reduced fertility, but from a population level, doing an essentially in-person clinical trial gives you much greater insights into the general widespread population level prevalence of disorders, hormonal imbalance, and therefore those are the things that we started to learn actually impact the numbers of patients who are going forward for fertility treatment. And what really our takeaway message from the numbers of individuals who were diagnosed through the test, the numbers of individuals who have at least one out of range hormone, is the questions we have coming out of this are: why are they not getting help sooner? What is wrong with our current method of assessing people? But furthermore, are the fundamental factors that lead to diagnostics even fit for purpose? And I'll talk about that in a second. So to start, I have learned more about regulation in setting up a company than I have almost any other aspect. And we were very dedicated and determined to making sure that we could hold our heads up high. But the first thing I'll say is that in regulating both an in vitro diagnostic kit, in regulating a clinical decision tool and a medical device, and in getting quality and care commission approval for not just testing, but screening, diagnostics, prescriptions, you realize that it's not a simple form that you will apply for any one of these things, but it's not only a changing landscape, but it's also a very confusing landscape. Furthermore, it's quite naturally a very expensive landscape. And what we've realized in doing this is that there are many other people who do not fully understand, but moreover do not care about what they're offering. And what we've seen across the landscape of reproductive health is that it is, especially in the context of fertility clinics, it's quite opportunistic and it's very competitive. And therefore, if one clinic is offering one thing, even though they may not actually agree with whether it's working or not, another clinic will offer it because customers will go to a competing clinic. But for us, I think having regulation that proves your diagnostic capability was very important. But I wish it had been straightforward. It's been a very long journey for us in doing that, and an upward battle. I think more clarity is certainly needed about what is needed, when it's needed, how it's needed, but also a complete revision. So how do we start to look at regulation outside of the UK? How do we start to look at making that regulation almost like the blueprint for scale across multiple jurisdictions and countries? And furthermore, when you're moving beyond just a diagnostic test, when you're moving into a realm where we will see post-pandemic, people not only want but expect some of their healthcare to be digital and remote, we now start to question, how do we revolutionize healthcare to become independent of the need for an in-clinic appointment. But how do we also regulate then the changing landscape whereby our clinical tools aren't necessarily people anymore, that our clinical tools are digital? And what's incredible to me is when we look at the investment that's gone into digitalization and technology in other essential aspects of life, one of which is our finances. And what's horrible is that $30 billion has been invested in the last few years in fintech, and only less than 100 million has been invested in femtech. And it shows you that while we need investment into this space, actually it's already on the back foot. And so if we can't get decent funding to turn around an area of health that is so radically in need of transformation, and I don't mean this from the perspective of the fact that diagnosis times take too long, but I also mean it from the practical perspective of the sheer volumes of patients that are requiring this and the numbers of individuals who are going forward for treatments. At the weekend, I spoke at a conference in reproductive medicine and spoke with somebody from the one of the largest IVF clinics in Europe. And they amazed me in saying that they do a transfer every 10 minutes all day. And when you think about the practical element of how are we revolutionizing key practices within the IVF clinic, sure, that's fine. But how do we actually start to look at changing the technology or introducing technology that enables accommodation of the sheer volume of individuals? And one of the areas that we're working on obviously is accommodating sheer volumes through digitalization, but it certainly does need a revisit when it comes to how we start to think about the volumes of patients and how do we transform, moreover, how do we regulate digital diagnostic tools? So the lessons I've learned when building a reproductive technology have been both from the lens of an academic, but also through the industrial and business and institutional questions that I've had. I've started to question some of the other industries, some of the businesses, some of the institutions that are built to create solutions. And some of the questions that I've had are around keeping up with the regulatory rules, those that are changing, those that changed post-Brexit. But moreover, how can innovation, which we know is can be catapulted outside of academia, how can innovation be supported and not limited by regulation? Because at present, it's everyone talks about regulation in terms of a regulatory landscape, and I would say that landscape is a minefield. The other questions that I've really started to question are the technology gaps. I think in terms of thinking practically, the example I gave, how do you physically transfer embryos into a woman every 10 minutes for an entire day? Those are things that are technology gaps. How do we start to address the underlying causes that are leading to the numbers of people? But mostly, how do we better communicate both the regulation and the technology to the people for whom this technology and regulation serves and protects? And the academic questions I've had are quite cynical, really. Who regulates research? When we build proprietary algorithms based on data, we have proof points. But when we look at research and when we start to build proof points around research, and we start to look at some of the diagnostic criteria and guidelines that have been used day in, day out by primary care providers, by secondary care providers, who creates those standards? One of the first things we did with building an at-home test that tests people's bloods was we looked at what are the symptoms. So we took all of the international diagnostic criteria and guidelines for the most common reproductive health conditions, 9 of them, and we said, what are the guidelines that a doctor uses to diagnose somebody with this condition? But there wasn't just one guideline in any one of those conditions. There were a number of different governing bodies. And so we started to create a table that enabled us to cross-reference what was the governing body, what was the evidence, the research evidence, what was the diagnostic criteria that was needed, and the combination of those diagnostic criteria points. And what we very quickly realized was, yes, we've got fintech for finances, and we've all got apps for our finances and to add and subtract and calculate our financial situation. But what do we have for our overall health? And what do we have in a burdened health system where you're relying on one individual to aid that individual? If combining the guidelines for just one reproductive condition took a myriad of symptoms of different factors in order to say whether somebody may or may not have a condition, how do we actually create a calculator? And so in creating essentially a calculator for reproductive health conditions, we then started to look at the other determinants. So when we test somebody's blood, let's look at the reference ranges. What were these reference ranges built on? Then we look at the lab. Who's responsible? In a provider to patient relationship, where the provider is suggesting that a patient gets a test and the results are determined by a lab, which sends them back to the doctor to convey this message. Who regulates this entire process, whereby we look at the reference ranges for an individual condition and we say, have these been set on a decent population? Have these been set on enough people for us to rely on reference ranges to say whether somebody is out of range or whether they fall within the reference range that we have created as a standard? And therefore, who is responsible for guidelines? Guidelines seem to be that that governing bodies come together, experts come together. They're not data-led, they are practitioner-led, where you look for patterns within individual patients, and they say the majority of these patients with X, Y, and Z equate to this. And therefore, when guidelines become unfit for purpose, who's responsible for that? When diagnosis times for endometriosis are 7 years, for PCOS are 3 to 4 years, that's a failing of the guideline and a failing of a system by which patients are relying. So those are the questions I've started to ask in and around that, and how we embed new research within clinical practice. And moreover, how can we build guidelines that are patient and data-led? So these are just some of the regulatory considerations or in and around data infrastructure and digital biomarkers, and therefore digital doctors.
We'll move on to the next speaker now, who is Dr. Alison Campbell, who is the Chief Scientific Officer and Director of Embryology at Fertility. Alison is also co-editor of the Atlas of Time-Lapse Embryology.
Yeah, I'm going to talk about one of my favourite subjects, time-lapse embryology and algorithms in the laboratory. A little bit to say about artificial intelligence, and hopefully get some interesting discussion going later on. So no commercial interests with any of the companies I'm going to talk about, any of the products. I'm a minor shareholder of Fertility. And it's actually Fertility's birthday, 25 years, so happy birthday to Pet. Last month, yeah, we're 25 years, and I've been with the company that long. So I just wanted to show this because it sets up the presentation quite well, because I'm going to talk quite a lot about data. And all of our clinics across the UK have a common platform to collect the data, which is why we've been able to develop some of the products that we have. And because the laboratories all work in a very similar way, it's enabled us to make some quite interesting tools. So going back to basics, really, if we talk briefly about static morphological assessments and the limitations of that. And we know that assessing human embryos, it's not very well standardized across IVF laboratories. It's quite subjective. There's a lot of evidence to show, and we've got our own to show that one embryologist might not see a blastocyst, there's some examples here, the same as another embryologist. And what most of the world is still doing is looking down the microscope at the embryos at various stages and making a quick assessment on its morphology, on the shape, on the form of the embryo. It's quite time dependent. We've got consensus guidelines to help us do that, to make sure that we do it at an optimal time to get the best information we can. But clinics seemingly now are starting to do less and less manual observations, because we're realizing, certainly with all the time-lapse publications, that we're not getting that much useful information from assessing the embryo on each day of development. And in fact, we're very much weighting the decision as to which embryo to choose, to the very final observation, which is quite often the blastocyst. So the picture, the video, actually, I should start playing that, on the right-hand side shows time-lapse imaging of a human embryo just going through fertilization now, and you'll see the embryo developing, so very much dynamic compared to the traditional static morphological assessment. So what's happening with these time-lapse devices are that they're imaging the embryo through multiple focal planes every 5 to 10 minutes, right from insemination, if it's ICSI, or fertilization, right through to embryo transfer, the blastocyst stage, quite often. So you can probably see from this video that we're getting so much more information about each developing embryo than we ever dreamt was possible, actually, and certainly much more than the static morphological assessment that is very, very commonplace. So we're actually obtaining more than 1,000 images within 24 hours. Compared with static morphology, we may get one observation every 24 hours, but usually less. So we're collating a huge amount of very interesting data. So our database at Fertility is rather enormous. We've been using time-lapse imaging for about 10 years now. And so we have more than 25,000 treatment cycles that we have undertaken time-lapse imaging of embryos. So bearing in mind, most patients have 5 to 10 embryos. You can just imagine the size of that data set. So quite a responsibility and potentially a gold mine of information that we can use to learn from, to research, to analyze, and to develop algorithms to help us choose the best embryo. And that's exactly what we've been doing for the last 10 years. And we started really simplistically, just using our basic biological knowledge and common sense when we're watching these videos. Well, this is not a normal mitosis. This is not what we expect to happen. This embryo has gone from 4 cells back to 3 cells. So we would deselect those if we had embryos that were developing in a more regular pattern. And eventually we felt we had enough data to compare those morphokinetics, the profile of the embryos which had implanted or resulted in a baby, with those that had not. And we saw that there was a significant difference in the morphokinetic profiles of those two groups. So time-lapse algorithms are in use in many clinics now across the world. And there are 3 types. We put them all really into 3 buckets. They're the ones that are integrated into the software. So if a clinic buys a time-lapse device, some of them can come with a pre-programmed algorithm or model. Relatively easy to use, but some limitations in that they're developed from a very large heterogeneous data set. Of course, the manufacturers don't have their own data, so they've got some arrangements and they've collected anonymized data in order to help develop their own integrated software. And there is some evidence that these are better than morphology alone. It makes sense that it would be. The next option are published algorithms. Many groups and clinics have published their own algorithms, like we did about 10 years ago. We started to publish some of our algorithms. And so clinics could then choose to program their time-lapse devices and their own software based on published algorithms. Lots to choose from. They could in theory retrospectively test the applicability of those published algorithms on their own data, which is a very sensible thing to do. But there's not much evidence to support that these published algorithms built in one clinic are transferable to another clinic. And then there's the in-house derived algorithm, which is what we've chosen to do because we had the capability and we had the scale. Took a lot of time and expertise, but we built the data set very quickly because of all the clinics doing the same thing with the same devices, same culture media, and annotating the time-lapse videos in the very strict and similar way. And we started really simplistically, just 2 or 3 morphokinetic variables, and now we've got quite sophisticated algorithms. And it's good to have the reassurance that these are built and validated in-house. So we're currently, our latest algorithm, it's the 6th iteration, and we call it CareMaps. The MAPS stands for Morphokinetic Algorithm to Predict Success. So in-house developed, and the current one built on over 6,000 transferred embryos where we know the live birth outcome, live birth positive or negative. So it looks a bit complicated, but if you just look at the pink bars on the graph, those were the output of the modelling. So these were the predicted values for live birth based on a big data set. And we've got a score from 1 to 10 based on the embryo's chance of becoming a baby, a live birth. And the purple bars are now our prospective use of the same algorithm. And so it's actual fetal heartbeat. We haven't got them all through to live birth yet. So you can see that the model is performing almost exactly as we'd hoped. Perfect line almost, going from score 1 with a very low chance of a live birth up to 10, which is around nearly 50% chance of a live birth. And this is irrespective of patient age. So we're really lucky to have it, and it's working beautifully at the moment. So it's not an artificial intelligence, it's a statistical algorithm. So if we talk about artificial intelligence briefly, and it's everywhere. It's in our lives, it's being introduced in all aspects of our lives, speech and handwriting recognition, we've got gaming, healthcare. So artificial intelligence is all around us. So large data accumulated from our clinics can be used, and we've now got something to share with you, that it can be used to help assess embryos and help select embryos. And we can see across the world now, and ESHRE, and this year and last year, the trend for abstracts associated with artificial intelligence is just so steep. We can see we've got oocyte assessment algorithms, sperm assessment and selection tools based on artificial intelligence, lots for embryo assessment. We've got protocol generation for patients, which medication should they have, what dose should they have based on their medical history. So it's exciting times, but it's a little bit daunting. There's a lot going on in this area. But it promises so much. It promises automation and standardization, which is a great thing. So workflow could be improved, reproducibility could be improved, the subjectivity that we see with embryo assessment shouldn't happen. This is much more objective, it's faster, hopefully should bring cost effectiveness and clinical effectiveness. So it's really exciting, I think, that we've got this, but it's a lot to get our heads around. So machine learning for auto annotation. So I explained how the embryologist annotates the time-lapse videos. Well, what we've developed now is an automatic machine learning model to do that task for us. So because of this vast data set, and we had actually almost 500 million images of embryos that we used to train this model. Of course, we didn't do it in ourselves, but we did it with a third party who are very experienced at machine learning. And we provided 63,000 fully annotated embryos, and we've trained a machine learning model on our own annotation data to automatically and rapidly annotate for us. So saving a lot of time, just starting to roll out and collect the data and compare with our own manual scores. And these data from the auto annotation can then feed into the statistical algorithm, CareMaps, and generate the same scores. So we're also doing a lot of testing to make sure we're getting the same performance. So the evolution of embryo selection, I think this is inevitable, from traditional morphology, static assessments, following guidelines, to time-lapse morphokinetic algorithms with the automatic image capture. Still not universally accepted though. It's there's still meta-analyses to show that it's not working in some people's hands. So I think it's very much reliant on the quality of the data that's going in, the annotation practice, quality assurance around that. And so perhaps still more evidence required to convince many users. And then we move to artificial intelligence. Already available, moving very quickly, reproducible, some benefits, clear benefits over time-lapse morphokinetic analysis and selection, and also traditional morphology. But with great power comes great fragility. So we have to tread carefully and make sure we work in a very robust manner. So just to conclude briefly, if I may, non-invasive embryo assessment methods, they're not going to change the embryo. It's a non-invasive way. Nothing changes. It's just there to try and help us get the best embryo selected first for transfer, to shorten the time to a live birth for our patients. But these assessment methods that are non-invasive, the time-lapse, the AI, they promise to improve accuracy and reproducibility. We know morphology is subjective. There are many time-lapse models available, some more effective than others, but the jury is actually still out largely over whether or not they're benefiting patients. So in-house process validation and KPI monitoring is absolutely crucial, with large, reliable, robust data. And with this, AI promises to be beneficial, I believe, for ART clinics and for our patients.
Our next speaker this evening is Dr. Andy Greenfield. He is Honorary Research Fellow at the University of Oxford's Nuffield Department of Women's and Reproductive Health. Andy is a member of the Human Tissue Authority and the Regulatory Horizons Council. Previously, he was a member of the Human Fertilisation and Embryology Authority and the Nuffield Council on Bioethics.
So my topic this evening is how should we regulate genome editing as an assisted reproductive technology? And I just want to say that any opinions I express this evening are mine and not necessarily those of any organisation with which I'm associated, which Sarah just kindly listed. And I have no financial declarations to make. So I was a member of this this commission that reported a couple of years ago on trying to elaborate a responsible clinical pathway to the use of heritable human genome editing. I'm going to call it heritable human genome editing rather than germline genome editing. I was invited to talk about the regulation of germline genome editing, and I don't like germline. I'm not entirely sure I know what a germline is. I'm pretty sure most people don't know what a germline is. But what we're really talking about, I think, is allowing people who want to have genetically related children to have those children free of a specified disease. So in a sense, what we're talking about is allowing people to have a germline, which does seem to me to be important. Now, one of the outputs of that report was that there is a parallel process that pertains to the introduction of genome editing into the clinic. So in orange, you can see what is here called societal considerations, but in fact, what they are is political processes that ultimately will confer legitimacy on anyone who wants or aims to use heritable human genome editing. And those political processes are something that to some extent are part of the context for this evening's meeting, because we're talking about the possibility of amending the Human Fertilisation and Embryology Act, which of course is going to require a political and democratic process. The other parallel path in blue is the development, scientific and clinical development of actual methodologies that can be used to safely and effectively introduce edits into the genome, probably of embryos, but it could be into the genome of gametes or of gamete precursor cells. And so if you introduce these edits into the appropriate type of cell, then the individual born as a product of that genome editing will have that edit, if it works well, in every cell of the body, including in their own germline, and thus they will be ultimately transmissible. So that's why it's called heritable. So I was asked to consider regulation, but one thing that should be clear at the moment is that we probably don't have, I'm pretty sure we don't have a safe and effective methodology. If anyone thinks we do, let me know. But certainly the last time I checked, we didn't. And we don't have a lawful a way of performing human genome editing in this country in a heritable fashion. So don't try this at home. So just to reiterate, in case anyone wants to misunderstand me, willfully or otherwise, I understand that we could only regulate the use of genome editing in assisted reproduction. I don't mean in embryo research, I mean in assisted reproduction, if it was lawful to do so, and it ain't. But imagine, so everything I say from now on, it has to be in an imagined possible world where we do somehow make heritable genome editing lawful. So I'm going to start by saying a few things about regulation. Some of what I say here should echo some of the things that we heard from Helen, although we didn't swap notes beforehand, but there will be points of contact. I'm a member of this organization called the Regulatory Horizons Council, and we produced a report back in June called Closing the Gap, and it was about trying to get out how we can use lots of well-meaning principles for innovation-friendly regulation, how we can put them into practice. There seems to be no shortage of innovation-friendly regulation principles. The question is, how do they actually work in practice? And that's what this report was about. But to give you an example of the kind of things we're talking about. So we know that regulation is important in contributing to building public trust, particularly in uses of new and controversial technologies. I mean, knowing that there's a new technology must conform to certain standards, knowing that there are specific use cases, knowing that there are redress mechanisms if something goes wrong, all of these are part of the process by which you build public confidence. And I take it that it's clear that we need that kind of public confidence whenever this kind of profound and new kind of technology comes along. And we also know, however, and this is something that Helen said, that the design and implementation of regulation can unduly hinder innovative new technologies. So as an organization, we're about trying to understand how regulation can be used to positively influence and alter the trajectory by which a technology enters into society, rather than simply unnecessarily and unduly hindering it. So that means that regulation will be taking into account not just the standard precautionary considerations about safety, but also will take into account impacts on innovation. And some of the principles that we identified and discussed in the report are as follows: that one thing we want regulation to do is to adopt a proportionate approach to risk and potential, i.e. not to be excessively precautionary, but also not to be excessively liberal either. We think it's important that regulation and regulators do embrace ethics. I certainly see no harm in regulators having ethical principles by which they judge their own activities, and for them to be explicit about that. And certainly public engagement is important, again, for reasons I've already outlined, that if we're in the domain of public policy, understanding what public attitudes are seems to be relatively important. Regulation must take account of commercial considerations, something else that Helen said. So we live in a capitalist economy, whether you like it or not, and innovators need to attract investment. And to some extent, regulation and regulators can crush that very possibility at a very early stage. And that's something that also needs to be considered. We need to consider alternative forms of regulation. Helen talked about standards and guidelines, and that's definitely something that we should be considering, rather than just hardwiring and codification of detailed regulatory frameworks into primary legislation. What other tools do we have in the toolkit? I'll say a bit more about that in a moment. Regulation needs to get the timing right. If you build regulatory frameworks too soon, you can crush a fledgling innovative technology at the outset. If you do it too late, of course, you can allow harms to occur that were foreseeable and should have been prevented. So the timing is important. And of course, we want openness. We want regulators to be open and to see that the status quo is almost certainly going to be inadequate. They need to grow in an ongoing fashion, continually evolving. So let me say a little bit more about this thing about alternative forms of regulation. And I want to say something about regulatory sandboxes. And the reason why I just thought I'd say something about this is it seems to be that the use of genome editing as an ART does raise questions of great complexity and also great subtlety. The second is not always evident when you read about this topic, but certainly complex issues, which is why the literature is replete with discussions of heritable human genome editing and what it may or may not do, certainly ethical discussions, scientific discussions. And because it is such it's a technology that poses questions about our future. It poses questions about how humans think about the way in which they want to control their future and the way in which they want to use control of biology to control human futures. These are profound questions, and it is controversial. And that means, it seems to me, it requires being dealt with care. That's one way of putting it, I think. A sandbox. Now, this seems to go from the sublime to the ridiculous. A sandbox approach is always associated with the Financial Conduct Authority, is the way in which fintech explored ways of doing innovative and different things in the fintech space. So it's considered to be a safe space in which businesses can introduce innovative products and services and business models and delivery mechanisms without immediately incurring all the normal regulatory consequences. So in this safe space, it's different. You're not taking a new product and rolling it out across the sector simultaneously. So in the case of genome editing, that would mean you're not rolling out the permissibility to use heritable genome editing to all clinics simultaneously in the UK. That would not be, it seems to be, necessarily a sensible approach. So what other things can we say about sandboxes? Well, they're a way of prototyping and testing new approaches, by which I mean not just new products and services, but also new ways of regulating, because the regulator won't necessarily know how to deal with these new emerging technologies, which are disruptive. They need a learning process as well as the people that are developing the technologies. We're talking about testing and trialing technologies, but not abstractly or intellectually or theoretically, but in a real world environment, which is, as again, it's a learning opportunity, but it does rely on this concept of anticipatory regulation, i.e. the idea that regulators should be happy to sometimes support the introduction of innovative new technologies, not simply to see their role to block. It's likely that sandbox approaches will allow policy to evolve over time, because there will be regular iterations and reviews, and it will be outcome based. It won't be based on what we expect to be the case. It will be based on what actually happens. In the case of IoT, I mean, I guess Alex might say something about this. I presume it would require revising the HFE Act. I don't think we could adopt a regulatory sandbox approach to anything at the moment, given the way the Act is structured, but I'm not a lawyer. But it probably also means giving greater discretion to the regulator in terms of how to configure such a sandbox program. But it certainly means probably a degree of collaboration with which perhaps we're not familiar, a degree of collaboration between the sector and the regulator. So in the context of heritable human genome editing, we're talking about essentially a small scale trial. Again, it's not necessarily the case that any clinic would be able to apply to have its license amended to perform heritable human genome editing. We're talking about, at the very outset, only a small scale trial. Now, there are all sorts of questions there about how you gain entry into this trial, which I can just ignore because I'm talking. There would have to be defined use cases. The report that I mentioned earlier spends many, many pages talking about what might be warranted, justified initial uses of HHGE. There would have to be close interaction between the providers and the regulator, ongoing close interaction. It's not open-ended. This is not a one and done. You don't just say, okay, guys, just go ahead and do it. And, hopefully we'll find out in 3 to 5 years what's going on. It's a continual process of dialogue. There has to be a pause switch and an off switch. The thing about the technologies we were talking about is they may not work. We may not anticipate things that can go wrong, but things can go wrong. So there's an off switch and there's a pause switch. One thing I want to say at this point is it seems to be really important that in this kind of sandbox environment, I think it's important that reputations are not at risk and at stake. We don't want to be in a position where someone says, this cannot fail. I've spent my whole career designing this particular methodology. It cannot fail. Or even the regulators saying, well, our reputation is at stake. Everyone says we're great. This cannot fail. That all has to be, I think, abandoned, that kind of thinking. This is to some extent high risk. It might fail. If it does fail, you either put the pause switch on or you switch it off, and you go away and you think again, and you come back with sandbox version 2, if that is justified based on how things have gone. Of course, if it all goes swimmingly well, wonderful. Then the question for the regulator is, how do we roll out this technology? In what fashion? But it has to be open. It has to be open to scrutiny. We want to get away again from this process that if we have a parliamentary debate about heritable human genome editing, and parliamentarians say we should make it lawful, that's the end of the public scrutiny, that's the end of the openness and transparency. No, that openness and transparency should persist right the way throughout the trial, so that people understand exactly what's going on. I realize that raises complexities. And this is, again, it is an opportunity for everyone to understand what would happen if we do roll out this technology. So obviously, there are some outstanding questions. I really wanted to avoid this evening sounding glib about this technology. There's nothing to be glib about in respect of this technology. It's powerful and its implications are profound. So there are other questions. Who gets to decide whether we have a safe and effective methodology? And I think there has to be an international dimension to that question and answering that question. What are the initial use cases? How do we recruit to such a trial? How do we make sure that informed consent is truly informed? Is disease prevention the only acceptable use? What about oligogenic and polygenic interventions? I'm obviously looking down the track. This is why it's a technology that needs great care, because it potentially could touch many human lives in future. We're not talking about niche applications here. And there are obviously a whole host of ethical questions concerning societal impacts and what would happen if human genome editing in the context of the ART clinic is normalized.
So Andy mentioned powerful and profound, which perhaps describes our final speaker this evening, Alex Denoon. Alex is a solicitor and partner at the firm Bristows. He's an expert in life sciences regulation. He works with clients to devise and implement regulatory strategies throughout the product life cycle of pharmaceuticals and medical devices, including in vitro diagnostics.
I don't really have any conflicts here. These are mainly personal views. Whilst I act for a huge variety of people in the life sciences sector, I'm certainly not representing any of their positions here. These are my personal views based on many years of working in the sector. So I guess in answer to the question, how should emerging reproductive technologies be regulated? I'd really rather not call them reproductive technologies. I'd rather call them technologies that are used in reproductive health. They certainly aren't technologies that reproduce themselves. And that helps, in my view, focus us on the existing frameworks we already have and whether or not we need to deviate from those existing frameworks or whether we can rely on those existing frameworks. So in my view, the majority of these emerging technologies, although Andy's talk was particularly interesting or particularly controversial because it doesn't fit into the frameworks, which I touch on later. But we already have a mosaic of regulatory frameworks, and that the vast majority of these technologies are likely to fit into. And my personal view is I would avoid any additional supervision or, for that matter, alternative routes to market for these particular technologies. But let me step through my rationale for that. The basic frameworks for most of these technologies are either going to be medical devices or medicines. So by way of example, an AI system to assist with the selection of an embryo or to identify the optimal time in a cycle is almost certainly as a medical device, as it's software used to control or support conception. You could also fit it into an in vitro diagnostic medical device if you wanted, on the basis that it's making a diagnosis or a prediction. There are a huge number of AI medical devices that have already been approved and are widely used in the NHS. So there's nothing particularly terrifying about the use of the medical device regulatory framework to approve AI systems. The biggest concern in the sector is probably the use of unauthorized devices. There's quite a lot of enthusiastic activity. And then just so that before we leave the medical device regulatory framework, it's worth noting that the European Commission proposes an additional AI regulation for AI systems, which would include medical devices. And this is intended to address data governance, data biases, and mandate algorithmic transparency. That will almost certainly end up applying to virtually all products used in the UK, notwithstanding Brexit, simply because very few people are going to develop products solely for use in Great Britain. But so we already have quite a framework, and it's getting tougher by virtue of this AI regulation that is coming through the European Parliament. And then in terms of medicines, where the technology is a medicinal product, i.e. it has a pharmacological, immunological, or metabolic mechanism of action, then it's regulated in this as a medicine. And this would apply to products that are administered to the mother, such as a conventional medicine, a stem cell product, a gene therapy product to treat the mother, or a product that was used to treat that was administered to the embryo in utero, such as a stem cell product, a conventional medicine, a gene therapy product, or a CRISPR vector. So we already have these two broad regulatory frameworks that would apply. And if we look at these two regulatory frameworks, as you can see from this table, which even I can't bring myself to read out, notwithstanding the fact I wrote it, you can see that the safety and utility of these products has to be proven in order to be able to get the product to market. There are some slight differences. In the context of clinical trials, the clinical studies to get these products approved, it's mandatory for medicines, and it will virtually always be required for medical devices, the kind of medical devices that we're talking about here. And those clinical studies themselves have ethical and regulatory approval or oversight, as the case may be. And all of these products are subject to mandatory post-market surveillance and reporting of the results of those post-market surveillance outcomes. So you can see we already have a really good framework to help fit these into. And my strong recommendation would be to try and fit these kind of technologies, or the products that will spin out of these particular technologies, into these frameworks. We even have compassionate use exemptions for an unauthorized version of the product, where in certain circumstances you can go to the MHRA and ask for an ethical approval. And it's also possible, which is kind of what Andy was getting at, I think, with some of his discussion about sandboxes and kind of conditional approvals. There are also mechanisms whereby you can get a temporary approval for a product where the regulator can see this is very exciting, but it's going to require additional data, or there's a pandemic, so we need the vaccine. And the regulator can step in and grant temporary approvals for unauthorized products where they are satisfied. And in my view, I don't really think that there's a particularly strong imperative to have something additional to these frameworks. I think they're well understood, well-trodden pathways. If we were to add much more to them, I think we'll generate uncertainty and delay progress here. And I think something more abbreviated than this is also going to be problematic. And I definitely don't see much demand for that in terms of kind of clients that I work for. Then turning to Andy's points about slightly more unusual interventions, revolutionary interventions, and how they might fit into the regulatory framework. If one were to think about an autologous graft of the mother's stem cells, let's assume she's got a placental insufficiency or something, and we want to take some of the mother's stem cells and return them to the mother to try and help with a placental insufficiency, and those cells haven't been substantially manipulated. Those products are not. A product like that is probably not regulated as either a medical device or a medicine. It would essentially be an autologous graft, a surgical procedure. But whilst they probably fall into a regulatory gap in terms of our discussions, in my view, this isn't really reproductive medicine. This is a medical intervention that is seeking to assist the mother. On the other hand, if the product was a substantially manipulated stem cell or something else to similarly treat a placental insufficiency, then it would be regulated as medicine. And then the issue that Andy was addressing, in vitro correction, for example, using a CRISPR vector of a genetic issue, so a heritable or a germline issue in an embryo intended for implantation. It is more problematic and doesn't fit into the existing regulatory frameworks. So it's currently illegal, 100%. I would agree with Andy in relation to that. And I think in terms of going forward, this could well fall within the purview of the HFEA and require licensing on a case-by-case basis. However, if I had the power to do so, I would say I'm not entirely convinced that the HFEA would be well equipped to think about quality, safety, efficacy, all the points that I made here. They don't really assess quality, safety, or efficacy. They don't really manage clinical trials. They don't know how to do post-market surveillance. They don't know how to talk about compassionate exemptions. They don't know how to deal with temporary approvals. That's not being critical. They're just not institutionally equipped to address those additional points. So I think if we were to go down if we were to try and foster the development of some of these therapies, if we were satisfied that there was an ethical imperative and an ethical justification, in my view, it should then be handed across to someone like the MHRA, who is actually equipped to understand these things. Now, whether we call it a conditional marketing authorization or an extensive clinical trial or some other framework, there are multiple frameworks that the MHRA in particular is familiar with to use in these particular contexts. And that would be what I would use. Now, I wholeheartedly concede that when all you've got's a hammer, and I'm a regulatory lawyer, a life sciences regulatory lawyer, and I deal with the MHRA all the time. When all you've got's a hammer, every problem looks like a nail. And when the MHRA is my solution for everything, that's what I've come with. So I've fallen into my own trap. But that being said, so if it ain't broke, don't fix it. I would say the majority of these emerging technologies for use in reproductive health can fit very well into the existing regulatory frameworks. Those frameworks responded with remarkable agility to the demands of the pandemic, reviewing dossiers, approving vaccines in record time. And the regulators took a lot of risk, a lot of responsibility on their shoulders. And I think that they need to be commended and supported for this. My temptation would be to lean on the existing structures rather than go in any other more radical direction, in part because those radical directions do not tend to promote innovation. In my experience, they tend to stifle it because they generate ambiguity. I would prefer limited HFEA involvement and definitely no additional independent approval or prohibition through the HFEA, except at that conceptual level as to whether or not this particular germline intervention would be permissible. And I also would strongly support the suggestion from my learned colleague, Julian Hitchcock's suggestion of a clinical technology review body rather than going to Parliament for permitted processes. I'm loath to think that the HFEA can turn its mind to quality, safety, efficacy, post-market surveillance, exemptions, all of the things on my table. I don't think the HFEA knows how to do. That isn't to say they can't do it. It's just they don't have the institutional competence to do those things. And that's why I would prefer, if we cross that particular Rubicon with that particular technology, which I think is a long way away, if we cross that particular Rubicon with that particular technology, I would prefer that it fit into a more conventional framework, such as medical devices, rather than the HFEA saying, okay, either this particular clinic for this particular condition or this particular condition at multiple clinics or whatever, that it can fit into a blessing from the HFEA. I just don't see how they can address everything else that's crucial in addition to the ethical construct.
Right. So one quick response there, if I'm allowed, that the kind of collaboration I was discussing in the sandbox area would involve collaboration between the regulators as well. See, I don't think the MHRA would know one end of an embryo from another, and probably wouldn't know that much about the kind of things that happen routinely in the IVF sector, or certainly in the human embryo research sector. So it would be a requirement for the HFEA to collaborate with, because we're talking about competency, aren't we? And whether they have the appropriate competency. So if that requires collaboration between regulators, great. In terms of expertise, yes, you need expertise on tap. the HFVA needs scientific and clinical expertise on tap. I'm sure the MHRA does. And the other thing I would say about the MHRA is, my experience is that lots of people think they're already swamped. They're absolutely swamped. They can't cope with the demand for their services in a number of different areas. And so we would need to greatly increase their resourcing.
The MHRA's budget should increase tenfold. I think they're an extraordinary asset for this country, particularly post-Brexit. But I think that the lion's share of the heavy lifting needs to be done for that kind of intervention. And I appreciate we've jumped into that particular intervention when there's lots of other ones that can get to market more readily. I would say the lion's share of that heavy lifting needs to be addressed by someone like the MHRA who knows how to deal with all of those constructs. They don't know anything about embryos in the same way they didn't know anything about chimeric antigen receptor T cells until they had to learn about chimeric antigen receptor T cells. They didn't know much about mRNA vaccines until they had to put their sensitive bits on the block and approve them. The fact that something is unusual doesn't mean that they shouldn't be addressing it. The fact that it's unusual means that you need to bring the best of breed together to actually resolve it, in my humble view.
If it's an important principle to get the timing right of the regulation, which makes complete sense, do you think we're on the back foot now with AI in our field, seeing as we've got prediction tools already publicly available?
I mean, I think you're right. There is a sense in which there's a degree of catch-up being played, but I think there's an urgency, let's put it that way. Just to plug the Regulatory Horizons Council, again, we have a report coming out quite soon on regulation of AI as a medical device, where we have worked closely with the MHRA on developing, some thoughts about how this could be done. But you're right, there's an urgency to get this done if the UK is to be, a leader in this field and show leadership in this field.
Okay, I'm going to move now to the questions from the audience. So we have one for you, Alison, first, from someone who's chosen to be anonymous. Your database is impressive and your algorithm seems to produce very promising results in terms of success. I believe it raises some ethical concerns though. Are patients charged to use time-lapse while data are collected? And who owns the data coming from the embryos of the individual patients?
Right. Well, no, we are getting a lot of data, and it is benefiting our patients in a positive way. Yes, we do charge extra for our algorithms. We've invested a lot in the hardware, the software, the technology, the time, a decade. We didn't charge anything until we demonstrated efficacy in our own hands. And that's the same with everything that we introduce. Data ownership, it's a big question. We use the data completely anonymously, the video data. It's not dissimilar, I guess, to ultrasound scan data, but maybe you're more likely to be able to identify a patient from an ultrasound scan, whereas from an embryo, it's completely non-identifiable. So yeah, we own the data, it's protected, we own the algorithms, it's our IP. So I hope that's answered the question.
Yes, I think so, Alison. Helen, I wonder if you wanted to come in on this, because it could be similar things that apply to Hertility about who owns the data that you're getting in.
So data is an interesting question in itself, because I think the word data has become a dirty word since companies like Facebook and Google, or more so Facebook and Meta, have started using them for, I guess, swaying different opinions. And that was definitely true when people decided to delete all of these apps because of the fear of what their data would be used for and the fear of targeting for advertising. And I think it's actually done a disservice to data in the terms of healthcare, where we see that actually data can be used so quickly and rapidly to improve patient outcomes and to improve people's ability to receive care. From our perspective, the data is obviously internally Hertility owned. We would never ever sell the data. The aspect of selling data, I think, is a big fear for many people. But when, as a company, you look at data protection, data regulation, and protecting your data, you take that very seriously. I think when it comes to healthcare data, you only get one chance, and it's of imperative thought when it comes to building something and an infrastructure that can be used both for research, but also could be used further downstream.
I mean, interestingly, GDPR one of the few things it doesn't do, despite its extraordinary breadth and ambition, one of the few things it doesn't do is commoditize data. It doesn't create ownership of data. It gives you rights in relation to data from which you can be identified, but it doesn't create proprietary rights in relation to data. So I always feel a little uncomfortable when we talk about ownership of data, because it doesn't actually fit particularly neatly into the frameworks. I think there are protections against misuse of data, and I think that's the better way to think about these frameworks. But that's just my bias, and I don't do enough data protection to express a firm view.
Thanks, Alex. So I'm going to move on to the next question now. Again, it's an anonymous one. If we add special or extra layers to regulation of fertility-related technologies, do we risk implying that fertility should be treated differently from other areas of health and medicine? So I'm going to go to Helen first, because I can see her nodding most vigorously.
Fertility is already treated differently. It's been treated differently since the very beginning. It's so many of the practices that are directly impacting or directly put into clinical practice were never tested. Even down to practices like ICSI, which was an accident, and all of a sudden, well, guess what? It was trial and error, or else trial and success, and continue going. Vitrification was almost brought in overnight, where it was seen to be effective and to work, and that's, in a strange way, there's two ways of looking at that. One is that it's scary that we're doing these in-person clinical trials. In another way, you think, is it more effective? If something works well, should you continue rather than testing and spending a long time bringing things to clinic? I don't know. It's why I think the point I made around regulation and fertility. I often say this when I talk about CRISPR, is that when you compare the regulatory landscape or minefield that we have right now, in terms of application of heritable genome editing, to what the application of the field of assisted reproduction went through, it's non-comparable. I mean, and if you were to today look at assisted reproduction and all of the technologies associated with it and try and regulate for that, many of them would not be regulated for.
IVF is the fertility is remarkably heavily regulated. I mean, in effect, there's a tax on every cycle. Every woman who undergoes a cycle, the clinic has to pay a fee to the HFEA. It's extraordinary. I can't think of any other sector where there's a tax on the intervention. It's bizarre. It's Byzantine. It's weirdly written. The interpretations that have been adopted, it's moved in a direction that I think was never intended by the original legislation, but what would I know? Now, I get why that was the case, but I think it is very heavily regulated at the moment. And that's why I would prefer that when we look at a lot of these revolutionary, emerging, extraordinary, fast-moving technologies, that we try and fit them into a framework that makes sense, that we're very familiar with medicines and medical devices, rather than thinking of it as particularly exotic or special that requires this extra attention. I accept there are ethical issues, but I think they can be addressed in a relatively straightforward way, and trying to turn existing regulators into something they're not particularly well equipped to be, I would avoid like the plague.
I completely disagree. So I think it is very exotic and very special. And that's because I had nothing to say about fertility treatment. You know, HHGE is not a fertility treatment. It's not even therapeutic. I mean, the individual who allegedly benefits from the intervention doesn't exist when the intervention begins. So that's not even therapy. Some would say it's not even medicine. I think it is medicine, though, in the same way that contraception is medicine, is also not therapeutic. So, you know, we can have a broad definition of medicine, but it's definitely a special kind of intervention. It has special kind of consequences for human society. And that means I think it's entirely reasonable that it should have special treatment. It's entirely reasonable that it should have special competencies associated with it.
Could not the same thing be said there about PGTM? That doesn't.
Yes, it can, and I would.
If you look at the application of genome editing under the same lens as the application of PGTM and the frameworks that you need to undergo as a patient, from a patient's perspective, applying for each individual condition, it's the same rulebook that you would use for genome editing.
I think for me, where the rubber hits the road there, though, is that it should be the HFEA regulates it as they do PGTM. But that's probably the only thing they have in common, is that they're both seeking to prevent the birth of children with specific monogenic conditions. That's what they both have in common. But that's possibly, all they have in common. I mean, there's a whole lot of science, as you well know, involved in genome editing that requires whole other different scientific competencies to assess.
Just for any of the audience that we might have lost, PGTM is pre-implantation genetic testing for monogenic conditions that some people might be more familiar with being called pre-implantation genetic diagnosis, PGD. It's all terribly confusing. Okay, I'm going to move on to the next question, from Richard Ash. And this is to Alex and Andy. In view of current knowledge and regulations, how would you view, from the ethical and regulatory point of view, the pioneering work of Professor Edwards and Steptoe in the first IVF cases? So we'll go to Andy first and then to Alex.
Yeah, I guess I'm being invited to say, would I have introduced some kind of special sandbox approach? Possibly I would have done, actually. But I don't think that would have precluded anything, because we would have still got Louise Brown and we would have still made our merry way onwards. It's only if it had gone disastrously wrong that the sandbox approach would have proved to be prescient, but it didn't, fortunately. So, yeah, I think that what they did was, like all scientists and all practitioners, they are products of their time. It's very difficult to go back and judge their activities based on our own standards. We're now absolutely swamped and surrounded by guidelines of an ethical nature and a regulatory nature, and we're used to it. We're creatures of regulation in ways that they weren't, certainly not in this sector. So, yeah, I'm certainly not, I wouldn't make any condemnation of what they did at all. I mean, thank God they did the work they did, I would say. But there are more than one way to regulate anything. And you'll notice, you have to remember that I did say I was putting forward an idea. I never told you it was the right thing to do. I was just putting forward an idea, which is why we have discussions like this.
Exactly. Alex?
I agree. I think that, thank goodness they did the work they did. It's an extraordinary boon to humanity that they did what they did. I don't think there's a clear objection on the current framework, even if we look back with our current hindsight and look back and sort of criticize it. I think it would be difficult for that to fall foul of any particular regulatory frameworks. Sorry, just to be clear, I'm entirely in agreement that if we're going to go down ex vivo use of CRISPR to correct heritable conditions, I think that should happen in a regulatory sandbox. I might use some slightly different terminology, but I would like that to be. I would like the ethical issues to be dealt with at the beginning and monitored throughout. But then I would prefer that we use the frameworks of probably the medicines regulatory framework to think about how we actually measure this, how we deal with quality, safety, efficacy, how we deal with reporting, how we deal with monitoring. It's going to be difficult because the output is a being that doesn't exist already. So it's going to be a very weird monitoring that's imposed there. But I'd rather that we use the medicines framework in the way in which we do with, for example, stem cells or other cell therapies.
Alison or Helen, did you want to come in on this question?
Only from the perspective that I think there's many lessons that can be learned along the way, and one thing that we assume is that this is going to be a governing body that leads this, rather than actually, when we look at examples, for example, with mitochondrial replacement therapy, this was something that began with the patients, that was driven by the patients and pushed forward by patients, so that it eventually reached a legislative point. And I think there's a lesson to be learned in that. When a technology becomes so well recognized for its efficacy, then actually it stands to reason that the patients start to say, what about me? And could this be used for me? The reason I say so is that I gave a talk this weekend on about genome editing in front of mostly embryologists and IVF practitioners. And it was strange to give that talk in front of an audience that saw immediately the clinical applicability and related to the patients for whom that would be really beneficial. So it was strange to not be talking in front of any bioethicists and to not be talking in front of any, theoretical or even lay audience who thought, what are the most extreme use cases of this technology that we can think of, and in fact actually be speaking to people who were thinking how many rounds of PGD or PGTM they had gone through for patients and gone back and said there are no embryos left. They were coming up to me saying, like, when can we do this? Can you bring this to our lab? And I was like, no, the last guy who did it is in jail. We gotta hold on. But it did kind of alarm for me when we start to become much more familiar with the success of a technology, then people will start to demand it. And I think the sooner we, whether it's an entirely different body or a combination where both HFEA and MHRA, have cross communication, something should be put together sooner rather than later.
We should move away from the idea that everyone has huge reputational risks at stake every time we try a new technology. That shouldn't be the case. That would suggest there's something wrong about the way in which we are introducing emerging technologies, which have a degree of uncertainty associated with them, into the clinic. And again, I want to reiterate, in case Alison is concerned, I'm saying we need to put, AI in embryo imaging through a sandbox. No, not at all, because I don't think it raises the same ethical issues. The only ethical issues it raises are the ones that she's already been quizzed about in terms of accessibility and payment, etc. But it doesn't raise those kind of profound questions about human futures that normalizing HHGE would do. So, do think there's a special case. Although normally I'm not into exceptionalism. I don't like making special cases, but every now and then I think you have to make a special case.
Okay, so I'm going to move on. We've got another question for you, Andy. Gosh, everyone wants to know about genome editing. So if HGGE were to take place in the regulatory sandbox, as you suggest, what would happen if a child is born, because this is about making people who will have to live with the consequences, who either still has the characteristic which was sought to be edited out, or a de novo mutation that was clinically significant to that child's health? Where would liability rest, and what kind of recompense should be due to the child, given that by design there will be no legal or regulatory framework to fall back on? So I think that's part for you, and then that horrible legal question for Alex, I think. Well, you might want to answer that as well, Andy, but I'm giving you an out.
Well, I mean, if I did attempt to answer, it would obviously not be from a position of any expertise, since I'm not a lawyer. Although I would imagine that we're not talking about abandoning regulatory frameworks. We're talking about, as Alex said, temporarily suspending certain features of those frameworks. And of course, how that is done would be a matter for a case-by-case kind of analysis, because you can also imagine this operating in the case of extending the 14-day rule. How do we go about extending the 14-day rule? Do we allow every single clinic in the UK to apply to culture beyond 14 days? Or do we have a sandbox approach where certain clinics with demonstrable skills and expertise go ahead and see what happens and let the HFEA learn how to regulate experiments that go beyond 14 days, that kind of thing. What would happen? I take it the de novo mutations happen. They happen in ART, and they happen in normal bog standard reproduction. I don't think we can attribute any blame to de novo mutations that simply happen, unless anyone can prove that culturing embryos causes a higher rate of de novo mutation. I don't think anyone's ever done that. So I don't think de novo mutation would be anyone's fault. It was just bad luck. if they had the condition which they had been edited to prevent, that would be rather unusual, because I take it that most people envisage HHGE working in combination with PGTM. So you would do your editing and then you would take a biopsy and you would look to see whether or not the editing had been successful. Since you can't know the genotype prior to editing, usually because you're editing at too early a stage, you check to see whether the editing had been successful and whether whether the disease genotype was absent, which means they would have transferred an embryo with the disease genotype. So something's gone wrong in that scenario. But clearly, if that happens, there's a serious problem with the methodology, either with the scientific methodology or the clinical methodology or something.
British courts have consistently refused to compensate for wrongful life. So a wrong-coloured baby created by a mistake in an IVF clinic, they refuse to compensate for it, and a child that is born with an issue that could have been addressed and was missed through identification. The wrongful life claims are simply not recognised in British courts. Long may that remain. It's a kind of revolting calculus to get into. On the other hand, if someone was to participate in a clinical trial and someone was harmed by virtue of their involvement in the clinical trial, then clinical trial compensation guidelines would step in and you'd be compensated for all losses without having to prove negligence. And so that's another example of why I prefer the existing frameworks to be used on a go forward. But liability is not an issue. It's simply something that British courts haven't been willing to come at in terms of this being a life that isn't worth having.
So there's also another complication, which is one that won't make anyone happy, which is that the literature is replete with discussions of the non-identity problem, which is a philosophical point about whether or not the intervention in question just chooses a distinct individual to be born rather than alters an individual that is going to be born anyway. So PGTM just selects among possible human beings to be born. So clearly identity there is not preserved, and the intervention all changes identity. Arguably, genome editing might not do that, but some people think that even genome editing alters identity. So when identity is altered, harm and benefit don't apply. That's the catch. Harm and benefit do not apply in cases where identity is altered by the intervention. So that's a big problem. You might have a very clever lawyer who says, but the non-identity problem, Parfit 78, means there could be no harm in this case. I mean, I wouldn't want to be in the court that day, but well, actually, I probably would. And I'm not a philosopher, so please go and consult your local philosopher for a proper account of the non-identity problem.
I would like to know what importance to the endometrium you give in your ART cycles. It appears from your lecture that not even to mention the presence of an optimal endometrium is shown. And then the next question, I'll bundle them together, is whether you've considered using polygenic risk scores for your embryo selection. So if you could tackle those, please, Alison.
Fine. Yeah, well, of course, the endometrium is critical. We need an optimal endometrium to transfer the embryo to. So with the algorithms, we've built it on data where we've got an implantation or we haven't. Where we have got an implantation, that data is the golden data because everything's perfect. But we have to accept that where we haven't got an implantation or a live birth, it may not be the embryo. So it's not a perfect data set. So I think that's probably the point we're getting at. And I accept that, because the embryo may be viable, but if the endometrium is not, then we will get a failed outcome, and that data will go to the failed outcome data. So yeah, that's true, and that's probably why the algorithms aren't giving us absolute certainty. There is always going to be an element of uncertainty with it, but it's the same with morphology as well. If you have a perfectly morphological grade AA embryo and the endometrium isn't optimal, then you're not going to get the implantation. We don't have the data, polygenic data. We don't have the genomic data in order to link anything to morphokinetics. And to be quite honest, I can't really get my head around that. We don't have the data to do it. We don't have the genomic information from our patients. But you might find it interesting that we do for the PGTA patients, so the aneuploidy, we've talked about PGTM, so PGTA, when we're looking for aneuploidy, we have the morphokinetic profiles of many euploid and aneuploid and mosaic aneuploid embryos. So we have built algorithms which predict ploidy or classify the risk of aneuploidy. So it's non-invasive, it's not absolute, but for patients or areas, regions that can't offer, the patients don't want PGTA, or it's not possible to do it for technical reasons or regulatory reasons, then that may be an alternative way to select embryos in a non-invasive way to look for the aneuploidy risk.
Does anybody else want to come in on polygenic risk scores, because they are quite a controversial topic in this area?
One thing I would say is that it seems to me there's a kind of conceptual issue that always bothers me about this, which is that obviously you can imagine a very, very large number of embryos that any couple could produce. I don't know how many you'd have to generate. I mean, it might be thousands. And in those thousands and thousands of embryos, you could imagine exploring the genetic space that to some extent predisposes you to whatever it is you're interested in, you know, diabetes, cardiovascular disease, and also non-disease traits. But it would require, you know, I'm not a mathematician, tens of thousands of embryos to truly explore that genetic space. And it's only by truly exploring that genetic space will you get the significant differences that you really want to justify selection. And of course, most women, Alison, how many embryos do most women produce?
10.
10, right?
Yeah.
So 10 is not going to allow you to do much of an exploration of that genetic space. So the chances there are going to be significant differences between, you know, the probabilities associated with disease predisposition between those embryos. Seems to me, just as a non-mathematician, as a non-polygenic human geneticist, I'm a mouse geneticist, in case you were wondering. You know, that doesn't seem to me a great survey of the genetic space. So that seems to already limit whatever the ethical concerns are about selection and about the target traits. That would seem to limit efficacy. But, you know, I'm sure I may well be wrong. And actually, humility is a good trait in this sector. So if I am wrong and anyone's listening, please tell me why I'm wrong. I'd like to know.
I wonder what your thoughts are about the state of current regulation. You made the point, Alex, if it ain't broke, but I'm not so sure current regulation is as fit for purpose as it could be. For example, the table you presented highlighted the fact that medical device regulation requires only that products show safety and performance rather than efficacy. Is that a problem? For example, we're already seeing research suggesting menstrual cycle tracking apps that have dubious accuracy.
Efficacy is a word we use in connection with medicines because they go through randomized, normally placebo-controlled, but not always clinical trials. No one wants to have a randomized placebo-controlled clinical trial in relation to a pacemaker. So we talk about whether or not the device does what it says it is claimed to do. So it performs in the way it is intended to, and people then have to make a therapeutic leap as to the clinical usefulness of it. It's a construct of the way in which we think about medical devices being physical things that operate in a particular mechanism of action, and medicines operate in another way. If you want to make particularly big claims about your medical device, you need particularly big evidence to support it. I don't want to get into it in detail because it's a horrifyingly complicated position, but if you want to say that this particular product will effectively deliver a cure or a particularly strong outcome, you'll have to have the evidence to back that up. Otherwise, you won't be allowed to get a CE mark for those particular claims. The claims themselves are regulated in the approval process. I don't think that there's a big efficacy performance concern here. I don't think that it's medical devices get away with a light touch. They get away. It's just a different perspective.
Some people think copper bracelets can cure their rheumatism, but commercially available copper bracelets are regulated in terms of basic things like ensuring they're not toxic or radioactive, that they're not regulated by bodies that regulate rheumatism medicine. By the same logic, if users of technology mistake a well-being or recreational technology for a medical technology, surely that doesn't mean you have to regulate as though those people are correct.
If a manufacturer wants to sell the bracelets or the magnets as a treatment or a cure for RA, then they have to get an approval for it, and it's a crime for them to sell them making those claims. But you're right. If it is a well-being app and someone uses it for improperly, or goes on a frolic of their own and applies it for a different purpose, that isn't the fault of the person who put the well-being app on the market.
Wording is very important. So you cannot say to somebody, unless you have this regulation, you think you have a diagnosis of. And there are many, I guess, screening companies that have to really skirt around their wording because of the application of an app or a report or results being considered actually a medical device, because somebody's actions will be impacted as a result of reading what it is that you're going to tell them.
There's one question that I sort of wanted to ask that I think really came up from Alex's presentation, but applies, I think, both to Alison and perhaps to Helen too. And that was, you talked to Alex about algorithmic transparency and that becoming mandatory. And I'd like to know a little bit more, if you could just in a nutshell describe what that is, in case anyone in the audience is not completely clear what algorithmic transparency may mean. And then if Alison and Helen can come in, and then we'll start to wrap up.
The idea with the AI regulation, the European Commission's legislative instrument to fix AI, is that it will, one of the multiple fixes will be anyone who's got an AI system for use, for example, in this particular space, would need to be able to explain how the algorithm works and changes over time, such that a person using it, whether they be a clinician or a patient or a combination thereof, understand what is happening in the black box. So you have to give them a lay person explanation. That's going to generate a huge amount of work for people trying to explain how this actually works. But the intention is, I get the intention, and the intention is, if you're going to have to trust a black box, you ought to have some understanding of what's happening in the black box.
Yeah, I can hop on there. No, yeah, it's the ideal, absolutely. But for the end user, so for me as an embryologist, for example, using an AI algorithm in the laboratory, I don't think it's possible for me to understand it. I mean, it's a little bit different for me because the algorithms that we've developed, I do understand. Even the most complicated ones, I've watched it develop, I've sense checked it, and I've explained it to my colleagues, and they understand it at the time. 6 weeks later, it's gone because they're sophisticated and they're complex. But if with the end users of iPhones, we don't understand how Siri works, no idea. So I think as long as they are decipherable by an expert, I think that's okay for me. I'm not such a control freak that I need to get into the black box and understand every detail, because I just don't think that's possible.
And Helen?
I think the point to be made is in around not just understanding the black box, but making sure that the black box is updated. What we've seen when we look at our reliance, so when you're creating an algorithm, you rely on truth data. You need truth data in order to form it. You need to say, is this as a result of the other? And when every single individual actually has predeterminants that will lead to a different cause of a disease, a different result in their bloods, then actually you have to start to question everything that the algorithm is being built on. And therefore, when new information comes to the fold, the algorithms need to be updated. And so that's, I think, what we've really looked into. I mentioned reference ranges for individual hormones. Is constantly monitoring the data to make sure that it is stratified. And we talk about algorithms and we say this is great, it's going to determine outcomes. But the reality is algorithms have to be actually data has to be stratified according to the different people for whom it's going to affect. And unless we have massive data sets thoroughly stratified, we have to actually agree that one algorithm isn't going to work for every single person. That actually what you need is multiple algorithms for each type of person, and updating our data to match those individual use cases is very important in terms of just revisiting data.